So we were talking about extensions to Mendel and variations to this. We talked about Mendel was looking at complete dominance, complete recessiveness. We talked about incomplete dominance. We talked about codominance. Now we'll get into some other additional considerations. We ended last time talking about the heterozygote advantage, situation where someone who inherits the normal, what we call two wild type alleles, two dominant alleles of the hemoglobin gene. That's probably everyone in this room here. I'm looking around, probably everyone here has the normal wild type. Gesundheit.
Sorry.
And if we were infected by the Plasmodium parasite, we really would have our immune system could defend us, but it would probably wreak havoc on us. And people with malaria are very lethargic. In the long run, it will lead to death. If someone inherits a mutant copy, one mutant copy, so they're a carrier, they'll probably function pretty well normally, but the red blood cells are more prone to sickling, and it actually provides a little bit of protection because the red blood cells turn over in the blood more often, so the parasite can be kept at a lower load in the blood. Someone inherits two mutant copies, generally not going to survive to reproduce. So you'd say it would be weeded out, but the advantage of being a carrier allows that allele to stay in the population. So we've been talking about dominant recessive relationships. There is one gene, we're going to call it color allele. It's the thing that determines most of the color in a rabbit. And there are 4 alleles, 4 different versions. So they'd be like brown eyes, blue eyes, green eyes, hazel eyes, or something like that. So here are the 4 different alleles. And I've put the genotypes up here that get you this color. This is called agouti, and it's your standard wild-type rabbit color. These alleles get you agouti. These alleles get you, what's this one called? Dark. Yeah, it's a variation on the color allele. It's dark. This is a variation on the color allele called light. And this is albino. And someone define the relationships of these to me. Is there a dominant allele? Are there codominant alleles?
There's codominant alleles and
There's codominant alleles and what? And incompletely dominant? Explain those relationships to me. You don't know.
It's a little complicated.
Yes, it is very complicated.
I just see the black nose and the black ears on the bus.
Black nose and black ears on the bunny? Look at the genotypes.
I don't think there's like one allele that only does ears, for instance.
So you don't think there's an allele that controls just ears?
Yeah.
Is there something that is dominant over everything? Is there something that's recessive to everything? The big C? So if you have big C and it doesn't matter what you have, you'll be what? Agouti. Agouti is spelled, I think some people call it chinchilla. Agouti is the right word. Right? So if you have any other allele, if you've got one capital C, you look wild type. Anything else? Is that right? So we've got a small c, ch, a small ch, and just a small c. So we've got all combinations of those, and that's wild type rabbit. Wild, out nature rabbit, right? Is there anything else we can say about these relationships?
The black one, if you have a CCH and no big, or big C, then you just
So if you have small c, ch, call that dark. Do you need anything else to get that? So what happens is, in this guy, what happens to the pigments? It's almost got the same amount of pigment. The pigments are just concentrated in spots. So it's more like flaky, unevenly distributed like that. Does that make sense? That's actually what happens, physiologically. What else do you need to get this color?
Doesn't matter.
It doesn't matter. Yeah. If you have a big C, it's going to be this, right? So by default, all of those are out. So if it's a small c with an H or a small c, you look like this. So we can actually start to do, we can say big C is dominant over C, CH, which is dominant over these guys, C, H, and little c. Can we do anything else with this?
Yeah. Little c is recessive to everything.
Little c is recessive to everything. Right. So if we have C with an H, it doesn't matter what the other one is, you'll get this. And in this case, this is a variation of this in which all the pigments are concentrated in the tips of the animal, the tail, the nose, and the ears. Isn't it crazy?
Is this real or is this like simple?
This is for real.
It looks like the CH little one has red eyes. Does it have red eyes or is that
No.
just? Okay. It's just a weird picture.
Yeah, this is albino. Yeah. This is albino. It has no pigment. It has red eyes. Little horrible buddy, gonna get you. Some of you know, some of you are like, what the hell is this? Okay. So if what you want to do is you want to look at the heterozygote. And if it looks like both, then you're just talking about incomplete dominant or codominant, depending on the particular situation. If it looks like just one, then you can identify one as dominant over the other, so we make out this relationship. Okay? Cool. Great. Let's talk about pleiotropic effects. Sometimes you have one gene, and it controls two different things. So Mendel noted this. If the plant has purple flowers, it has green pods. If it has white flowers, it has yellow pods. And he noticed some traits were always linked like this. Some things, if it was this, it was always going to be this also. And one gene controls flower color and also pod color. It kind of makes sense. There are traits that we'll see another trait, and this is a for real trait later, and we'll see in mice, where coat color also controls being alive or not being alive. Lethal. We'll see an example of that in a second. So one gene having many forms or having many effects on morphology. Lethality is quite often where we see these things. So here's an example of this. I take an agouti mouse, pure breeding, and I take a yellow mouse, and we can't get a pure breeding yellow mouse, and I'll explain why in a second. But if I cross a pure breeding agouti with a yellow mouse, we can actually map this out in a simple Punnett square. This is agouti. This is yellow. That's the genotype of these guys. We get agouti, agouti, yellow, yellow. What's the genotype ratio? 1:1? It's the phenotype ratio. 1:1. What's the difference between genotype and phenotype? I hope you know.
Manifestation of the characteristics versus the genetics.
Yeah. What the DNA, what the code of DNA is genotype. Phenotype is what it looks like. Okay. If we take and do a different cross, if we cross a yellow to a yellow, check this out. It's up there, but I'm going to draw my own. All right, so we get this guy's agouti. What color is this? Yellow. Yeah, we have yellow. It's yellow. What color is this? Yellow. And this guy's super yellow, right? Or dead. In this case, it's dead. It develops just a little bit in the embryo and then it dies. It doesn't make it to survive. It's a lethal combination.
Why would it cause it to die? Like, what would that affect to cause him to die?
I don't know what else it affects. Sorry. Oops. What's the genotype ratio of this cross? 2 to 1. We're missing one of these guys. What's the phenotype ratio? 2 to 1. Right. Okay. Good. What is the color relationship? For hair color, which is dominant, yellow or agouti? Yellow? Why do you say that?
Because it's just a big A, and so big A's and yellow is Judy.
Say that again.
So the big A is Judy.
The big A is agouti, yep.
Big A with a Y is yellow.
So the heterozygote,
Yeah, is yellow.
so you look to the heterozygote, right? So in the heterozygote, the yellow is dominant over agouti, right? It looks yellow. For survival, of being alive or not being alive, which is dominant? Agouti? In the heterozygote, you're alive. If you're homozygous, yellow, you're not alive. If you have just one copy of the agouti, you're alive, right? If you don't have any copies of agouti, you're dead. Agouti is dominant over yellow in the heterozygote, right? Okay, that's all I'm going to do to help you recognize this stuff. Some of you look puzzled. Some look around like a lot of you are. How do you get that? Encourage you to go and practice with colleagues. Okay. We talk about gene interactions. This is sort of a counterpoint to the pleiotropic effects, where one gene has effect on 2 different traits. In the pleiotropic effects, you have 2 genes that affect one trait. So there are many, many characteristics in humans that we can identify this. Here's a classic example. It has to do with the morphology or the phenotype of the comb on the chicken, on the hen, on the rooster. The comb being this thing here, right? This thing. There are 4 comb types. There's 2 different genes. And the genes are in different chromosomes. So we have chromosome, I don't know what, we'll say it's chromosome 1, where there's the R gene. We have a different chromosome altogether. And there's the P gene is on this one. Okay? So what do? They do a crossing here. Capital R is obviously dominant to little r, capital P is dominant to little p. These guys are co-dominant. So here, and they do this, here's another example with the P's, but basically, they start with something which is capital A, little b, little b, little a, capital B, so we get all heterozygotes when we cross, right? It's heterozygote for gene A and it's heterozygote for gene B, right? So all of those guys are brown. Isn't that crazy? You start with tan, you start with gray, you cross them together and you get a third color altogether different. If it's a monohybrid cross, you'd say, oh, it's either yellow, it's either tan or it's gray, right? You can imagine why this would really screw people up. They go, oh yeah, blending occurred, right? Tan and gray, you put them together, you get brown, blending occurred. And if you don't pursue it down to another generation, you'd walk away with that mentality, right? Now when we cross these together, because we get recombination, we get our parental back. We get capital A, little b, so this guy does in fact look like the parent. And we get little a, capital B, and we get this parent back. But we also get this same as the F1 here. The F2 has the same as the F1. And that would actually be one of these guys. Right there. It's technically genotypically the same. But we actually get green. We get a totally new color out of this. So the effect basically is through the recombination, we never had capital A, or we never had the double recessive together, right? In the heterozygote, we wound up with this mix like this. And when you self-cross these guys, you can get a new recombinant that never existed before. And these guys apparently appear green. Another classic example is in the chickens here, in the hens. You can get 4 different phenotypes. 1, 2, 3, 4. But it's a 9:3 to 3:1 ratio. It's just a dihybrid cross. So in humans, there are multiple genes that affect eye color. And you can have people who have, you'd say, pure breeding brown and pure breeding brown that can actually have a kid with blue eyes. They've had generations of brown and generations of brown, and then they get blue eyes because there's another gene that comes into play. Let's do this in blood type. Blood type is a perfect set of genes to follow. Let me get to this right now. We'll get to the blood type in a second. Here's another dihybrid cross. I want you to check this out. We have a pure breeding flower. It's white, pure breeding. And that's in my lab here in the US. Someone over in Europe has their own pure breeding strain of white flowers. So we take these together, and the way we test the pure breeding, right? So we cross them together, they should get white flowers, right? Cross them together and we get purple flowers. I've gone 20 generations and never spit a purple flower out. This person's gone 20 generations and never spit a purple flower out. We cross ours together just one time. Every flower in the F1 is purple. Whoa, that's crazy. If we take that and cross that, we wind up getting a 9:7 ratio. How does 9:7 break down? Can you break that down for me? 9:3 to 3:1. Yeah, 9:7 is this. 9, 3 to 3 to 1. These guys group together are my 9:7. It's a there are 2 genes that cooperate for flower color. Let me show you why. You're probably saying, like, how the hell does that happen? It's called complementary gene action. Let me show you what happens here. Think of it as a biochemical pathway. I have some compound. All my markers are dying on me here. Okay. We process this enzyme A. Processes this. I'm just totally making this compound up. And we'll add a hydroxyl in there. Let's just say we do that. So this is no color. It's a colorless compound. When we add this on here, it's still a no color compound. And then we have enzyme B. And this guy maybe converts some methyl into something. Right? And this is now purple. This compound is purple. Color. So what we have in the cross, mine is pure breeding. I have little a, big B, big B. And if I self-cross these, I have a recessive or a dead non-functional A gene. My enzyme doesn't fold up right. The person over in Europe has capital A's but little b's. So this enzyme is dead. This enzyme is non-functional. That's what I mean. Change the dead for non-functional. So if I have this as my pure breeding strain, and my guy can't make enzyme 1, I can't get this reaction to occur. So if this chemical is produced in the plant, it can never get processed into the purple compound. The person over in Europe has a dead version of enzyme B. The enzyme B is not functional. So that plant can make this compound, but it's still no color, and it can't finish the job. Does that make sense? So when we cross them together to get the heterozygote, one capital A has to come here, and one capital B has to come here, and a little a and a little b. This guy has to get one of these and one of these, one of these and one of these, so that has to be the genotype. And the organism now has half an active number set of enzymes here, and half a set of enzymes here. So it has a little bit of enzyme A, and a little bit of enzyme B, and it's going to make some of the purple compound. Got it? So we wind up with a heterozygote instead of the main reactions happen. It's called complementary gene action. This is a fundamental concept we're going to explore more when we talk about the cell cycle in really great detail. This type of, your understanding on this complementary gene action is fundamental to understand a complementation assay, which is one of the most powerful ways of determining what genes are responsible for what activities in a living organism. People studying yeast, the cell cycle, cell division in yeast, using this assay in the space of 5 years, probably did between 50 and 100 years worth of research if a cell biologist like me had been doing it. It was incredibly powerful what they did. Totally advanced your understanding of cancer. Complementary gene action. Yes?
So that's just what is that term referring to? What part of everything's just explained?
One gene complements the action of another. This gene can make this chemical, but it doesn't really have any noticeable effect. This gene can make this reaction happen, but it doesn't have any noticeable effect. You put them together, you get a bigger effect than you could have predicted before. They complement, and you get some greater effect. All right, let's talk about epistatic interactions. And this is, again, a dihydrogen cross. And we're going to go back to the blood type here, the blood typing. So we talked before how we have this H factor. And on that H factor, you can have no sugars, blood type O. You can have sugars that are covalently connected in the A way, you have A-type blood. Where the sugars can be covalently connected differently. And that pattern is the B-type, so it's B-type blood. And it was discovered there is yet another blood type up there. This was discovered over in India. It's called the Bombay phenotype. And let me tell you the situation. I don't know the exact details on this, but here's a rough approximation of this. Knowing what we talked about last time, we have a female. She is type O blood. Right? And the male is type A blood. No, he's type AB. Make it simple. Okay? What is his genotype based on what we talked about before? What? Capital I, A, capital I, B. What's her blood type? Little I, little I. Okay? This woman had a child by this father. She's married to this guy. And the child is type O blood. What is that child's genotype?
Either somebody lied or something weird happened.
So it had to get a little I here. So the child has to be little I, because it's type O, right? And so this guy donated his little I from somewhere, right?
Sure.
So obviously, she's been cheating on him, right? Because the kid must be either A or B. Because the little I had to come from somewhere. So these are real cases that happen in India, and at the time this happened, I don't know the details on that society over there, but apparently women have a certain role. At that time, they had a certain role. And if a woman were to cheat on a husband like that, she could be cast out of the family. You know, I think become something like an untouchable or something, and have no support network. And it was actually reasonable, apparently in the times, that the man would be entitled to do awful things to the female physically. Kill her, do bad things to the body. What was eventually discovered is, if they looked at the genotype of this woman is actually but blood type O. And so that is when they discovered this thing called H factor. That's a protein. That's a gene. So if you have your messenger RNA, probably almost everyone in this room, you got your ATG, you get your termination codon, you make your H factor. This woman, for that gene, an ATG and then a termination codon right after it. So she is unable to make the H factor protein. She has the enzyme to produce the sugars that are covalently connected in the A format, in the A pattern. And she also has the gene to make the sugars in the B form. She doesn't have anything to stick those sugars on. The sugars get made and then they go floating away. And type O blood is defined as no sugars on the surface. So this is type O blood. This is what she is. She's not even type O blood. But in a blood type test, she looks O. Does that make sense? So she is this. She can't make H factor. So let's say the kid is this. The kid has to get a little H from here. What blood type should the kid be? AB. The kid should be AB. The kid's blood type O. What is the genotype of the father? The father is big H, little h. Why not little h, little h?
Because he has
He'd be O. If he had little h, he wouldn't show his AB. So he gave a little h here. And this kid looks O, even though the kid is AB at the I allele, at the IG. Does that make sense? So epistatic, one gene masks the effects of the other. Epistatic. Yeah.
How often does this happen, and does it not give an effect on blood transfusions? And
Yeah, so let's talk about that. How often does it happen? It's a mutation that appears to happen one time in India. And in India, if you have money, you want to keep the money in the family, so cousins routinely marry, so the mutation got back upon itself many, many times and expanded in those populations. So depending on which human population, which subgroup of humans you talk about, it can be very, very rare, or it can be basically non-existent. So in the populations you find it, it's almost not there. Okay. What happens? What happens to this person if this person gets type O blood? They reject it because the body says, that's not my shape. I don't do that on the outside of my blood cells. If you give this person type O blood, they are dead.
So is there nothing we can give them?
You can give them,
If they have a bank.
you can give them not even O blood.
If
You need to find someone who's also Bombay factor.
Can they donate to the O and the other one?
Oh yeah, if these guys are universal, these are omni-universal. These guys can donate to anybody anything. Because they have nothing on the surface. They don't look like anything. Does that make sense? They're the super universal donors.
Was this a problem with India?
Yes. The things that happened to those women were inhumane. If this is the person, if it's me, if this is me, I'm donating blood to some bank, and I don't care how much it costs. They're keeping that stuff in storage so that if I'm in a car accident, there's blood for me.
How'd you know if it's you?
Get blood tested. If you type O, you can go get tested for the Bombay phenotype. You are so unlikely to be Bombay phenotype that it's. Do you guys hear about that guy who's like a white supremacist fella? And he had a blood test done, he's like 1/16th African American, 1/16th, Black American. Did you hear that? It happened all like on one of these like Jerry Springer type shows. He's like, oh, they're diluting us all out and kind of stuff. And then the test came back, and somewhere in his lineage, a couple generations ago, some African. So he's like 1/16th Black. It's kind of hard to come to grips with that, right? So blood typing is awesome. We get dominant recessive relationships going on here. We get co-dominant, so we get big I, little I is your typical completely dominant. We get co-dominant relationships. We get multiple genes acting on a single phenotype. So we get epistatic interactions. Might see some of these using a blood example on the exam, right? There'll be real-world examples based on this, real-world possible combinations. I'm going to ask you, what's the genotype of this person? What's the phenotype of this person? Is this possible? All right? I want you to think. So go online, and there's all kinds of blood genetics problems you can solve online. Okay. Let's talk about penetrance and expressivity. If I muddied the waters for you a little bit, but you're still like, okay, it follows genetics, it follows Mendelian genetics. Okay, I can do a monohybrid cross between the stuff and kind of work through it, right? Yes? Okay, let me get this really, really messy. Mendel was really, really lucky. He looked at things that are completely dominant, completely recessive. If you have pink flowers, if you inherit a pink flower allele, you're going to be pink, period. There are circumstances, in humans especially, where maybe you're a heterozygote. You're supposed to have maybe let's just make one up. If you're heterozygote at the A gene, say you have a dark spot on your back by your kidney, right? So if you're that or that, you have a dark spot on your back over each kidney. There are circumstances in humans where you inherit this. You're the heterozygote, so you should have that dot, but for some reason you don't make it, and we don't know why. Your genetics are not being expressed. So we get into this question, first of all, anyone know who this guy is? Anything notice something interesting about this guy? I have to turn the lights out here, sorry.
Extra finger.
Here's his thumb, and he has 1, 2, 3, 4, 5 fingers. He has 5 fingers and a thumb. What's that called? Polydactyly. Does anyone know what that hat is from?
Expos.
Montreal Expos. This is a major league pitcher. He's retired now. Alfonseca, Antonio Alfonseca. I think that's this guy. He was a relief pitcher. Has an extra finger. Actually uses it in one of his pitches. He uses it to grab the ball and help him get a different spin on the ball. Interesting. Polydactyly. Turns out it's dominant. I'll turn the lights on. So, anyone in here have an extra finger? I've been surprised by the PKU. Anyone here have an extra finger? No? We're all these little recessive, non-5, non-6-fingered person. That's all of us, probably. So he, Alfonseca, is this. So polydactyly is dominant, right? What percent of his children will have an extra finger?
Does his wife have an extra finger?
Huh?
Does his wife have an extra finger too?
No. 50%, yes. So the cross is this. This is his wife, and this is Alfonseca. So half of his kids are going to inherit this, and half of them will have an extra finger, right? Worked for him. Advantageous, right? He was making millions of dollars a year. Natural selection at work. It turns out you can actually inherit the allele for the extra finger, and for some reason it's not expressed. Genotypically this, but you phenotypically look like this. Penetrance and expressivity. I'm going to show you this. So, penetrance is penetrance for population. Penetrance for population. P, penetrance, P, population, right? Penetrance is, if you have 100 people of this, you would expect 100 people would have the extra finger, right? If you actually have this, 95 of the people have an extra finger. 5 people don't show it. 5 people don't express, or sorry, 5 people don't have that. The whole population of those people doesn't show it. Okay? I'm really screwing things up. In most cases, we have no idea why. So that's penetrance. Here's an example. Here's a person, this is a gene called RB. I hope some of you are familiar with this gene called RB. If you're wild type and mutant, so you're a heterozygote, you got a wild type copy and a mutant copy, you are 75% likely to get cancer in one of your eyes. If you're homozygous and recessive, so you have 2 mutant copies, you have non-functional, 99 out of 100 of those people will have cancer in their eyes, okay, in eye cancer. 1 out of 100 isn't going to get it, even though genotypically they're in this class. They're in the high-risk cancer group. About 1% of them don't. So it's not absolutely 100% penetrant in the population. Let's talk about expressivity. Expressivity is individual. Penetrant population, expressivity individual. Penetrant population, expressivity individual. So let's look at expressivity. Expressivity is how intense it is in one person. Expressivity is here. Of the 75% that are going to get cancer, some people just get cancer in one eye, some people get cancer in both eyes. How intense it is. Is it just one eye or is it both eyes? Okay? In the single individual. So we go back to polydactyly. Some people who have the extra finger, Some people who don't have the finger, they actually have bone structure for it in their palm, but they don't have the finger projecting out. Some people that have polydactyly, they've got a full-on pinky hanging off there. That's totally functional and usable. I think his was, I want you to imagine you broke your finger and or you really stubbed your finger real hard and you can't really bend it very easily. I think his was like that. He had limited motion in it, with limited ability to use it. The expressivity in a person is very different. It could be a whole finger poking out there or a little stub like he had. Questions between penetrance and expressivity? You can affect the expressivity in yourself. Here I've given an example, and these numbers are not exact, but they're pretty close. Someone who's a carrier for a mutation in the RB gene has a 75% chance to get cancer in an eye. If that person decides to smoke, they can change that number such that they go up to 95% likely to get cancer in an eye. The environment that they're encountering can augment or increase the penetrance in the population if everybody smoked. In addition, they're more likely to get it in 2 eyes than just 1 eye. So what affects whether the finger's going to poke out really far or not? Probably the nutrient level when they were in the uterus of their mother, chemicals they encountered, all kinds of things. Random chance. Yeah.
So do they have like the full bone and tendon structure for a functional finger?
I think it depends on person to person.
But it can happen. It could be like a completely.
I think it's not a fully functional like my pinky. My pinky isn't totally functional. I can't bend it really on its own, right?
Okay.
I think it
But it's
can
not
be
just
pretty.
like a protrusion of bone. It can have some movement. It's not
Yes.
just awkward bone mass.
Yes, and I've never seen someone like this. It may be that they're controlling a lot of it from down here.
Mhm.
But in general, I mean, the code for it is your body's just going to say, oh, well, we got another one of these here. It's another kind of segmentation pattern thing.
Okay.
Could be on both hands.
Yeah, it'd be both on hands. Yeah, they could have on one hand and not the other, potentially. In the old days, they used to cut them off. You're
What
not
about
normal.
toes? Or is it just hands?
Hands and toes, yeah, both.
Oh, so it affects both?
Yeah, yeah.
Okay.
The arms.
I don't know. Questions like that. The question is, I mean, this gets into questions about what's normal. They used to cut it off because that's not normal, right? But I think maybe he had better baseball skills in general than I did. But if you ask him, I'm not normal, right? You think about this stuff when we're talking about genetic engineering. Who's going to make decisions about, is someone with Down syndrome not normal? Should people be saying, we should make sure that nobody ever gets that 3rd chromosome and is Down syndrome? Because that's not normal. Is blue eyes normal? Is brown eyes not normal? Anyone with brown eyes should be eliminated as embryo. So we get this pure and beautiful blue eyes. Steel blue or gray blue like mine, right? Okay, we are a full-on lecture behind. Let's wrap up with this other one. We've talked about proteins that fold up. I think I introduced this before when we talked about the Arctic fox. There are cats like this also. So the cat has the enzyme, but if you increase the temperature, that thermal activity, in the cells, the protein falls apart and denatures. So the enzyme used to make the pigment is not functional. But in the parts of the cat that are out away from the central core, so the central core of my body here is warmer than the temperature outside of my body out here. My extremities are cooler. At this temperature, the enzyme won't denature. So the enzyme stays intact and makes the pigment. So you can get creatures where the extremities are all dark in color, the ears and the nose here, but the core of the body is light in color. So I guess in theory, if you took this cat and you threw it in the sauna and left it there for a couple weeks, you come back, the whole thing would be white, right? Or well, white. It would be all white because you would have denatured all the enzymes here. I almost said the same thing as you. That's the kind of thing on it. It'd say, oh, you take one of these cat. What's this cat called? Is this a Siamese cat or something like that? Fluffy cat.
Very scientific.
So on the exam, if I ask about fluffy cats, that's what we're talking about. Because again, you take one of these cats and you throw it in the Arctic Ocean for a couple of weeks and bring it out. What color is it? Explain to me the genetics behind this, right? Now, I'd probably say you take a cat and it lives outside in a cold environment and its color has changed or its color's not changed or something like this. And tell me what's going on. What's the genetics? So those are called temperature-sensitive mutations. And I want to introduce that term. Complementary gene actions allows you to map out a pathway, right? Many genes that all contribute to one effect, and temperature-sensitive mutants. Both of those are absolutely essential for the complementation axis for cancer research needs. Okay, we're a full day behind. Just remind you guys, Friday I'm out of town. We're not meeting for lab this week. We have class on Monday. Yes, this Monday we do. Right. So we'll get started on the PCR on Monday. See you guys on Friday.
Are you required? Yes. Can you possibly take my paper? I have an interview. Sure. Wherever.